Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium, and I want to welcome you to our webinar series. Today, we're joined by Frédéric Choulet from INRAE, and he is a group leader there and a lead in bioinformatics. He's been an essential part of a lot of the IWGSC efforts from the very beginning, primarily working both with the 3B genome sequence, which laid the foundation for the IWGSC strategy, all the way through to the reference sequences and all the versions of the reference sequences. So it's a pleasure to have Fred with us today. But before we get started, I'd like to give just a brief overview of the IWGSC. We are an international consortium and we have more than 3,400 members. 918 institutes or companies are engaged with us in some way, are participating in activities, and we're in 72 countries. We also have 10 sponsors, and I want to take this moment to thank them for their support. They make it possible for us to have these kinds of webinars and also to try to coordinate activities globally and activities related specifically to reference wheat genomes and annotation. So our vision post the release of the reference sequence is to continue working towards tools that will enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. So our next webinar will be the 17th of November, and it will be focused on yield gene cloning in wheat, something that has become increasingly possible now that we have reference sequences. So you can register for that webinar already, and I would encourage you to do so. So just to let you know, this webinar will be recorded and posted on the IWGSC YouTube channel shortly. You can subscribe to the channel so you never miss an upload. The presentation itself will be followed by a Q&A session. You can submit your questions in the Q&A panel already, and as we go through, you can submit them. We may not get to everything, but Fred has agreed to answer anything we don't get to in writing, so the responses will be made available on our website and on the YouTube channel. Please monitor the chat panel and you can see messages from the organizers, and that may include links to papers that are referenced in the presentation. I believe already you can download the handouts from today's webinar, both my presentation as well as Fred Choulet. So without further ado, let's turn it over to Frédéric Choulet, who's going to be talking to us about the sequencing the genome of the French wheat variety Renan. So Fred, thank you for joining us today, and I look forward to your presentation.
Thank you very much, Kelly, for the introduction. And thank you for the invitation to talk. I try to share my screen. Just tell me if it's okay for you.
You're in, so you're good.
Yeah. You see the slides moving? I switched.
Great.
Okay.
Yeah.
Thank you. Yes. So today I have two parts of my talk. So since we published the Renan genome paper, I have a first part summarizing how we reach a high quality reference sequence for RNA. And my second part is more about transposon dynamics, because this is a topic I like and I push for years now, and we have novel results about this. So this will be my second part. But first of all, Renan. Renan is a French bred with cultivar, widely grown, especially for organic farming, especially because it has durable resistances for many fungal diseases. These genes of resistance are mainly carried by introgressed segments, introgressed from a wild species, that is a genus ventricosa. So ventricosa is a tetraploid species with an N subgenome and a D subgenome. And a segment of 2N has been introgressed on the 2A in Renan, and a region of the 7D chromosome has been replaced here by the 7D of ventricosa. And the 7D segments carry the resistant genes to high spot called PCH1, and other genes of resistance are known to be carried by the 2N segments, the YR17, LR37, SR38, and CR5. So I won't talk about gene cloning today because the paper I mentioned here is a paper that is a paper about the genome sequence, how we assemble the genome sequence, how we annotate the sequence. So it's more technical than a gene cloning paper. Anyway, I start my presentation with always with slides with the the main vocabularies again for genome assembly and technologies. So basically, the workflow for getting a reference genome now is to get reads from sequencing technology, then assemble into contigs, scaffolds, and then finally is to get one chromosome sequence for each chromosome. This is called a pseudomolecule. Okay. In the wheat community, we have experienced many advances and revolutions in those all along this workflow. First by using Illumina to get high quality assembly. It was the time where we were assembling the China Spring, so the reference accession in wheat. It was published in 2018, and the revolution was mainly the step with the de novo magic assembler. But since that time, many advances have been made, especially the Bionano technology for scaffolding, high throughput scaffolding, the high-C technology for organizing the scaffolds along chromosomes, and of course the new technologies that allows to get long reads. So obviously most of you knows that now there are, let's say, two technologies called PacBio and Oxford Nanopore Technologies to get long reads. At the time we began the project of sequencing the Renan genomes, it was not clear which of these technology would be the best one, and in collaboration with the Genoscope, we have decided to use the nanopore technologies. But definitely things may be different now, I would say, because PacBio is extremely powerful. But anyway, I will present you the hybrid strategy that makes nanopore reads, Illumina reads, Bionano maps, IC maps to get the Renan genome assemblies. And before talking about Renan, I have a slide with where we are now in the wheat genomics, how many genomes are available in September 2022. Since Chinese Spring, in fact, we have many others genotypes with a high quality, reference quality genome assemblies. In 2020, 10 genomes were released by the 10+ genome projects with NR gene-based, so Illumina-based genome assemblies. Then the Zhang 1817s, also with NR genes, was published in 2020. Filder wheat, which is a variety used for transformation, especially. Carriega, also, I think it was last year. The Renan is the only one using the ONT technology. You see now, most of the novel ones are done with the PacBio Wi-Fi technologies. And I also have seen this year, Attraction is also a new variety sequenced by the group of Martin Machin. And you can see the papers now. It's not papers focused on genome sequencing, but it's mostly papers that focused on identifying gene cloning for resistance, and the genome assembly is within these papers. And we have also many other triticums and echelon species with a genome sequence, a high quality genome sequence available. Urartu, Durum, Decucuhides, Tauschiae, plus 5 other genomes that were sequenced with Nanopore and published last year. It's the phytopsis sections, but now I will focus on Renan and how we achieved the assembly of the Renan genome. So it was published in GigaScience this year. It's in collaboration with the Genoscope, the team of Jean-Marc Horry, and the team of Arnaud Bellec and CNRGV Toulouse, INRAE, mainly. So we used a combination of Nanopore and Illumina. So we produced 30x Illumina reads plus 60x depths of coverage with Nanopore, but we selected only the longer reads to reach 30x depths of coverage for the assembly. And just to give you an idea, if we consider only reads larger than 50 kb, we have a 15x depths of coverage. So we have really long reads with the Nanopore technology. But the problem was at the beginning of the project, the error rate in the reads, because if you know the Nanopore technologies, you have the strand of DNA go through the pores, gives you an electric signal that is base called into nucleotides. And at the beginning, it was 3 to 4 years ago, let's say. We had 12% of error rates in the reads and 4% in the assembly. So the group of Jean-Marc Horry at the Genoscope made a lot of improvement, especially to improve the base calling and the polishing. And we finally got a high quality genome after many rounds of polishing using luminaries, especially. So what you do is you map the reads and you correct the errors in the assembly because it is covered by a 30x luminaries here. And I do not go through all details, but this is an histogram just to give you an example of how the polishing was important. Here you have the raw assembly before polishing and after polishing. Okay, and we have used here markers coming from Chinese Spring that we mapped on the Renan genomes. And what we see is that at the beginning, we have only a few percent of those markers that are completely identical between Chinese Spring and Renan at some percent identity, 100% identities. And after polishing, half of those markers are completely identical between Chinese and Renan, which was much more close to the expected, what we expected. Okay, and only 10% of marker missing, which is expected when you know the presence-absence variations between Renan and Chinese Spring. So yeah, polishing was really important, and if you use Nanopore reads to assemble the wheat genome, this is absolutely necessary. Okay, don't try to be too long. Bionano is something really useful, and it's including into every sequencing project. So we build Bionano maps, use optical maps to organize the contigs. The contigs are in blue, and to build scaffolds. This is something really usual now. These are the metrics. We were able to assemble a genome that is 14.3 gigs. So it's really close to the other genotypes. Generally, when we assemble now a wheat genome, we got a genome size between 14.1 and 14.7 gigs. And we obtained 2,900 scaffolds, so approximately a bit more than 100 scaffolds per chromosome. In terms of quality assessment, we proved that we were really close to the completeness because 98% of the genes known in Chinese Spring were found in our assembly, and 92% of the HiSBIPs, so the markers coming from the TE space, the transposons, are found also in the essences, very close to completeness. And there's a tiny difference in terms of quality value, so the accuracy of the nucleotide sequence in the assembly. We are close to 99.95%, which is a bit lower than what you could get with the on-gene-based assemblies, like 99.99, because it's only based on Illumina reads. Okay, next step was to organize those scaffolds into 21 pseudomolecules. And actually, if you do this completely de novo, maybe I could zoom on the map. Yes. You could see that you have many, well, for those of you who knows, who have already seen Hi-C map, what you expect to see is a complete diagonal, red diagonal. But here you see many red dots outside the diagonal, which means you have inconsistencies between the Hi-C data, which means that probably you put together scaffolds that are not close to each other. Okay, so it was not efficient enough to build 21 chromosomes from the 3,000 scaffolds here. So we needed to adapt the way we build the chromosome sequences. And in fact, what we did is to guide the anchoring of the scaffolds based on China Spring before running the Hi-C corrections. And to guide an assembly based on the synteny with Chinese Spring, we did not use genes because genes are not, you have not enough genes to do that because, okay, sometimes you have scaffolds carrying genes, but sometimes not because genes are lost in the notion of transposable elements. So to anchor the scaffolds, we use ISBP markers. ISBP markers are small tags that target junctions between transposable elements. And the good things with ISBP is that you can map them uniquely on the whole genome. It exists only one locus for these ISBPs. And you have one ISBP every 3 kb, so all your scaffolds would contain many ISBPs. So we did this. We guided the assembly, the ordering of the scaffolds of Renan based on Chinese Spring, and just by doing this without any Hi-C data, you can see that just by doing this, you are able to get something really, really clean, except a few inversions like this one, and on chromosome 2A, much more inversion that we need to check with the Hi-C data. And this is what we did. And it was much, much better in terms of Hi-C map. In fact, we have a much clearer diagonal in red. And we have seen also that, okay, there are some mistakes like here. There are some corrections to make, and this was made in my group by Pauline Lasserre. And it was feasible because now it was made by hands using the juicer pipeline. You can see here an example where you have a scaffold that you should place here. Okay, you have something not clean here. A scaffold on chromosome 5b that could be on chromosome 5d. This has been done for every cases of inconsistencies on the map. So we corrected 18 chimeric scaffolds, for example, and we changed the position or orientation of close to 200 scaffolds. So 10%, let's say, of the assembly was incorrect at that time. And we ended up with 21 chromosomes for Renan, and less than 1% of the scaffolds that are unanchored and put into a chromosome unknown. And I show you here the dots plot, so before Hi-C correction, and after Hi-C corrections. Okay, and you can see that all these things that were inconsistent between 2A of Renan and Chinese spring were corrected here. And this is an inversion that is a true inversion validated by IC data. Those inversions are usual when you compare with genomes. Here you can see a dot plot comparisons of 2A from Renan with the 10 genomes released by the 10+ Genome project. And you see that you have some inversions like this and this that are sometimes shared between different accessions and sometimes specific. We found in actually only 2 inversions that are specific to the Renan genomes compared to these 10 other genomes. I would say the good point with nanopore reads and nanopore assembly is that you get very large contexts compared to the NR gene-based assembly. So here is an example of a region containing an important gene. This region is close to 2 megabases, and it's assembled into 2 contexts in Renan and to many, many more contexts in the other genomes available. Okay. And so this is one of the main advantages of using nanopore. We have a conjugate 50 that is close to 2 megabases. I think it's 2.2 megabases. So it's really, really large. And another good point with using long reads is that you can assemble complex motifs like microsatellites. And we gave this example in the paper of omega gliadin genes. You have an omega gliadin gene cluster on chromosome 1b. In fact, in the reference sequence of Chinese spring, all these genes were only partially assembled because you have a stretch of ends that is due to the fact that you have a big microsatellite within the gene, in the coding part of the gene. So in Renan, it's perfectly assembled and you have no ends in these genes. So these are 2 main good points with the long reads nanopore. Okay, let's switch to gene annotation because I think assembling a weak genome now is not a question, a research question. Let's say it's something that you can do. We are now in the area where either with PacBio or Nanopore, it's absolutely feasible to have a really high quality genome sequence. The problem is more about annotation, because predicting gene is really not trivial. It's really not something easy. And I would like to remind the people that choose the Chinese spring reference genome annotation, for example, that we produce this annotation in a collaboration between 3 annotation groups. My group in Clermont, the group of Manuel Spanager in PGSP Munich, and David Sporbreak in Aram Institute. And by a huge effort trying to reach the highest quality possible, we ended up with 107,000 genes, what we call the high confidence genes, okay, IC genes that people use for analysis. But I would like to remind that it's the limits behind sometimes that we are not confident that a gene exists. Sometimes it's really hard to say that the gene exists. What's its structure, the exon intron structure? And I would like to remind the fact that there are more low confidence genes than high confidence genes in the reference annotation of Chinese spring. So just to say that it's really hard to say if something is a gene or not. Okay, we have more than 160,000 low confidence genes. And just to give you examples of why this is so complicated, it's because, okay, we have here a case where it's extremely easy to annotate the genes because this gene here share high level of similarity with many proteins known in sorghum, in ardium, brachypodium, rice. Okay, it's even, you have evidence of transcriptions based on RNA-seq data that you have 1 exons, 2 exons, 3. Quite easy, you can run a program, predict the genes is a high confidence one. But in many cases, it's not that clear, okay? You have only partial evidence that it's looks like something coding, looks like sheer similarity at the protein level, only few evidence of transcription. So sometimes it's really difficult to decide if a gene exists or not. Okay, and to give you a good example for that is that years ago, we had this 3B chromosome that has been used for a long time as a model for genomics. 3B was sequenced in 3 different initiatives by the back-by-back approach, by the in the TGAC-V1 in the chromosome survey sequence, for those who knows. But anyway, it's 3 different approaches to assemble this 3B, annotated by 3 different groups. And you can see that just by the number of genes, it's a bit different, okay? Just the number of genes, sometimes it's really different. But if you compare genes that have been annotated by different groups, the similarity between the gene catalogs is really low, okay? Only 26% of these genes predicted in 3B were identical to these ones, okay? And it's only 12% here when you try to cross-validate the 3B genes in this initiative with this one. So just to say that de novo gene annotation is complicated, okay? It's extremely complicated, and depending on the resource you use, and de novo annotation is not adapted to pangenomics, okay? This is the message. And every time we will go for another genome or another with genomes, I hope that we don't not go to a de novo annotation, because it creates many differences that do not really exist. It's just methodological differences. Okay, so obviously what you want to do when you have a new genome like the Renan genome is not to annotate de novo from scratch, okay? Is you want to make gene projections, or you can call this gene projections or annotation transfer from a reference. So obviously the reference is the China Spring annotation, and you want to map the genes in the Renan, the novel assembly of a new genotype. And usually you can do this with, people do this with GMAP, which is a classical software for mapping a CDS on a genome. But in fact, when you do this, when we started doing this with Renan, we found 12% ambiguous mapping. It means 12,000 genes that do not map correctly. So we could not correct manually 12,000 genes. So we tried to improve this, because the GMAP versus the whole genome introduced misalignment errors. Okay? This was at the origin of the pipeline that we developed in-house called MAGATT, or Marker Assisted Gene Annotation Transfer for Rtbs. And it was developed by Helen Hamber in my group. It's a snake-made pipeline. And the ideas behind this pipeline is to target first the region where you predict the gene to be. So, because the problem with wheat is that every time you want to map a gene, this gene is many times present in 3 different copies, plus the paralogous copies, okay? And many times you have problems mapping genes. So what we will consider is the environment in transposable ions, okay? Because transposons around make a signature that you could use. Using ISBP markers, you can predict where is this gene in a Renan, okay? So MAGATT will take the ISBP markers on the 3' and 5' of the genes, map them to Renan, predict the target regions containing the genes, and compute the alignment in a very tiny, small interval, okay, where you are sure you have no problem of mapping these genes. This is what MAGATT does. It maps the genes, recalculate positions of CDS exons, and using MAGATT, we were able to find 98% of the Chinese spring genes, okay? So it was much better than the 12% of ambiguous mapping. And finally, we go to de novo annotation of only regions that were not present, not shared with Chinese spring. Okay? And we found 4% of Renan-specific genes, representing close to 5,500 genes. And these specific genes, the genes specific to Renan, are mainly carried by the introgressed segments. Let's zoom on the introgressions because I want to talk a bit about the introgressions, of course. So remember, we know we have two introgressions in the Renan genome, one on chromosome 2A and one on chromosome 7. First one on 2A, we know that we have an introgression of the chromosome 2N, which is a different subgenome. It doesn't share similarity between N and A. It's really two different subgenomes, okay, in terms of sequence similarity. But if you align the subtelomeric region, so 40 megabases in Renan, this is the telomere here on the left, Renan compared to Chinese spring. What you could do, obviously, with this software called Rtbs comparison tool, it's a very old software, but it's quite cool. You can see many similarity regions. It's because it carries transposons. Transposons are repeated. It generates match everywhere all along these regions. But if you increase the stringency, you can directly see that, okay, the end of chromosome 2A is different from the 2A of Chinese spring. Okay, and you can zoom on this region here where the similarity stops. Okay, and this is the way we have identified the breakpoints between chromosome 2A and the segments of chromosome 2N coming from ventricosa. You zoom here, what you would see is that, in fact, it's the recombination, because this integration occurred by homologous recombination. The event of recombination most probably occurs within a gene, a big genes with more than 10 exons. And it creates an hybrid genes between ventricosa and estival. Okay, these integrations carry a bit more than 500 genes. Okay, and approximately 20 to 25% of the genes are specific to ventricosa compared to estival. But I would not detail here the complete comparative genomics of this region. Okay, let's zoom on the other, the 7D, the second introgression. Here is the telomere in Renan, the telomere in Chinese spring. The size of the region is close to the size of chromosome 2A, and it's approximately 30, 40 megabases, carrying 500 genes. Okay, but 7D from ventricosa is much more close to 7D of estival compared to N and A. Okay, you can see that you have synteny, conserved regions. Okay, you can even see that the end of the chromosome is inverted between what comes from ventricosa, what comes from estival, and it's not obvious to see where the introgression point is, but it's probably here, and you can zoom here and see that okay, you move from a region highly conserved to something less conserved. And if you zoom again, you can see that you switch from something conserved at more than 99% identity, so it means this is estival versus estival, and here it's less conserved. You switch to 97%, 98%, which means it's probably a different species here, and this is probably the segments coming from ventricosa. And again, the integration recombination events probably occurs within this gene here. And that's all what I will say about the genome assembly of Renan and the characterization of the integration. And I move to my second topic. I hope I'm not too late, but I would like to talk about TE dynamics and the new results we have about that. It's because we published several papers for a long time now. It started with Josquin Daron, who was a PhD student with me, who has developed new methodologies to annotate transposons in the wheat genome. We published 4 years ago with Thomas Vicker and Ivan Goodland a good story, an interesting story comparing ABD subgenomes of Chinese spring. I will say a word about that. And we continue with Romain de Oliveira, who was a PhD student with me, trying to go deeper in characterizing the structural variations that affect transposable elements, but using resequencing data. It's hard using reads. Now we are in the area where we have access to fully assembled genomes. That's why it's important to move from resequencing data to analyze all these genomes. And this is the topic of Nathan, the PhD thesis of Nathan Papon, who is handling his PhD with me. And I will talk about new results we have about that. So first start with just showing you what the genome, the wheat genome looks like. You have genes here, but genes only represent 1 to 2% of the genome. The rest is made of transposons. Transposons inserted into each other's. It's the fast evolving fraction of the genome, and we would like to characterize better the variability of this fraction of the genome. But for that, you need adapted tools, especially to annotate transposons, because annotating transposon is a hard task. And this is what we developed years ago with Josquin. Okay, so we started by classifying all transposons known in weeds. It represented more than 5,000 known transposable elements. We classified them by families and subfamilies. This is extremely important when you want to study dynamics of transposons, is to know what are the relationships between subfamilies, families, and superfamilies. Otherwise, it's really hard to understand the dynamics, because you believe that two transposons that do not have the same name are completely different. But in fact, no, they are related. And this is what is important to know. This is why we reclassify from scratch the non-transposons to build a new library called Clarity Rep. Okay, and Clarity is a software pipeline we developed here in Clermont in order to improve the quality of the row annotation, the row similarity search with transposable element databases, because what you would get is something like this, something with predictions overlapping. And Clarity did this improvement in order to get something clean, where you can see that you have a transposons inserted into this blue one. Okay, so this makes the difference to have access to this type of information, high quality information, compared to this. When we compared the TE content of A, B, and D subgenomes of Chinese spring, we had really surprising data. We had surprising results. So it's a paper we published with Thomas Vicker and Idrun. What we have seen is that ABD have been subject to a near complete TE turnover, meaning that when you compare ABD, you can only see that genes are conserved. Intergenic space is absolutely not conserved, but it is shaped by the same families of transposable elements. Okay, so what you could expect if you believe that transposons are not subject to selection pressure, they are subject to silencing and bursts sometimes. What you could expect is to have ancestral TE families evolving independently in ABD lineages. Some may amplify, some may disappear, and at the end, in the hexaploid genome, you may have big differences between ABD. This is what you could expect, but this is absolutely not the case. When you look at the abundancy of different families, what you would get is a 1/3, 1/3, 1/3 proportions of every families okay, almost every family. So you have something balanced, you have balanced proportions. So, and no families specific to one lineage. This was really surprising, and we concluded that in fact, families tend to maintain in balanced proportions independently in diploids. Okay, this was surprising, but this was true. And when you do a tree on a given family, you can see that A copies are related, are phylogenetically related, B copies also, D copies also, as if they amplified independently within diploids before polyploidization. Okay, and sometimes you can see even rare events of amplification within the tetraploids AB. So every data, every results tend to make us conclude that families present, T families present in the ancestors maintained. Okay, it's completely changed. Okay, it has a turnover, a complete turnover through million years of evolution. But at the end, families maintained at a certain equilibrium. Okay, and that's we also raised the hypothesis that there's probably a structural role of TEs likely under selection pressure. And the first layer of evidence for that was that distance between genes tend to be conserved. Okay, not the content. Okay, it could be completely different transposon here and here, but distance between genes tended to be conserved. Okay, really, really interesting in terms of dynamics of transposable elements. So I continue working on that aspects, especially with Nathan. And what we did with Nathan is trying to confirm those hypotheses. So not by comparing ABD, because the limit when you compare ABD is that nothing is conserved, okay, in terms of orthology between transposons. What you can do now is, because we have many genomes, many high-quality genomes, is to make whole genome alignments of B genomes together, A genomes together, D genomes together, so that you can see recent events of insertions and deletions, and you can confirm the hypothesis we raised by comparing ABD. So I just will give you, if I have times, but please can tell me if too late, but if I have 5 minutes, I will just show you these results. So what materials did we use? We used every species with a high quality genome assembly, so like Rtbs, Durum, Urartu, Tauschi, and 13 different ST1, 13 different genotypes of ST1. We aligned A genomes together, B and D. Okay. The strategy to do that, it's really complicated, because you cannot assemble gigabases of repeats like this just by using BLAST. Okay, we selected only, well, we searched for collinear genes, but in many cases genes are collinear along the 3C genomes. Okay, but if you select collinear genes, you can select intergenic spaces, intergenic regions that are collinear and orthologous between those species, ST1 and Urartu, for example. And we split the genomes into thousands of intergenic regions, and then restricted the BLAST alignment to individual regions, so that we can have clean alignments, okay, and see which regions is inserted or deleted, where are the presence-absence variations. Let's say. Okay. We did this for the whole genomes of these species and identifying what I call the conserved fractions versus the variable fractions of the genome. These are the results. Okay, this is first estimation of what is variable when you compare ST1s to Urartu, Hydes, Durums, and other ST1s. Okay, here you have the percentage of what is variable. In fact, it's around 6% of the A subgenomes is variable at the intraspecific level, a bit more when you switch to interspecific level, and really high when you compare to Urartu, which is more distance phylogenetically. And I will not give you details of all numbers, but what you could see is that the variability of the transposable elements is between 5 and 15% of the genome. Okay, 5 to 15% of the genome is variable when you compare two different genomes, 2A, 2B, or 2D genomes. And again, as it has been seen with SNPs, when we can see that B genomes exhibit a bit more variability than A and that D being the less diverged, the less polymorphic genome. And the other interesting thing is that we didn't see any genome expansion or contraction. When you see the recent events of insertion deletions, we did not see any expansion or contraction because the percentage you could see in Chinese Spring is similar to the queried genome. Okay, it's 776698. It means that the variable part is quite similar in terms of size in these genomes, which maintain the genome sizing maintains during the TE turnover. And we go deeper and try to identify which families have recently transposed, okay, by identifying those elements that corresponds to an event of presence-absence variations with extremities of a given element. We did this for whole genomes, and we find between 4,000 to 13,000 copies of novel insertions. So it's the larger sample of novel insertions identified for those complex genomes. Okay. It's a lot, but it's only represent less than 1% of the genome, okay? Because we have 4 million of transposable elements in a weak genome. But interestingly, what you could see is that the number of transposition events, the recent transposition events, the numbers are very similar when you compare Chinese Spring to a query genome. Okay, you have 11, for example, with Dicocoides, 11,000 novel transpositions that are elements present in Dicocoides, absent from Chinese Spring, and the same present in Chinese Spring, absent in Dicocoides. So it means that the turnover, the amplification of transposable element is stable in all these lineages. It means that there's no more transposition in the hexaploid than in the diploids. This is not what we have seen. So what's the impact of polyploidy on TE amplification? I would say none, okay? Transposition looks stable in all these lineages. And how many families were active recently? This is also surprising, but almost all of them. We have traces of transposition for 346 families, representing almost all families known in the wheat genome. So this was really surprising because people always believe that you have TE burst of a few families that make the genomes diversifying. This is absolutely not the case for wheat. Okay, this is really my last slide. I hope I'm not too late, but it's just a zoom on the 20 most abundant TE families, okay? And this is the relative abundances in the ABD subgenomes, okay? If you look at the novel insertions, it's not the most abundant families that transpose more, okay? It's not correlated. Like you have novel insertions of all these families, but it's not really correlated with the abundance of the family in the genome. But what's interesting is this is a comparison of Chinese Spring with Tao Xian, so based on the D genome, okay? These are novel insertions, novel copies present in Chinese Spring, absent in Tao Xian. And this histogram is the reverse. Novel insertions of TE element present in Chinese Spring, absent in Chinese Spring. And you could see it's completely the same profile, meaning that these families transpose it seems it's transposed at stable rate, okay, in all these lineages, as if it's at an equilibrium, okay? And it's the same for A and B subgenomes when you compare Chinese swing with Durum, for example. So this was really surprising, but this is in line with what we have concluded when we compared ABD and concluded that it seems that there's a strong equilibrium of these genomes instead of a meth of TE burst completely different in the ABD lineages. Okay. My conclusion from this part is that we didn't see any genome expansion contractions due to TE transposition, no impact of polyploidy, no TE burst. All families were active recently. Surprising, but this is what we found. At a transposition rate that is stable per family. Okay, it's like family have a stable transposition rate in the recent history. So equilibrium for the genome and probably the role of TEs to maintain a genome architecture probably under evolutionary constraints. But I will not say too much about that, and I would like to end up now with thanking all the collaborators and my group in Clermont. All the names are represented here. I hope not too long. Thank you for listening.
Thank you very much, Fred, for really an interesting perspective on a genome sequence, first of all, and specifically one from a variety of wheat. So we do have a lot of questions coming in, and as I mentioned earlier, please feel free to put your questions in the Q&A panel. Even if we do not get to them today, they will be answered in writing subsequently. So we have the first question, which is probably the most direct, is based on your experience, do you recommend PacBio or Nanopore for initial long-read sequencing?
Okay, I like this question. Now, obviously, it's clear that PacBio has made really strong advances, and we have evidence that it works pretty well to assemble large contigs without errors. It's true that at the time we began this project, it was really unclear, and the Genoscope has the technology in hand, has nanopore technology in hand, and would like to experience that on a complex genomes. But it was a really hard task, trying to optimize every programs for polishing. And so we made it. Okay, it was not easy. We made it, and we're happy with that. But definitely, PacBio is much more simple because you have no errors in the what we call the hi-fi reads, and you get now less than 10 contigs per chromosomes with PacBio. So I do not recommend anything, okay? Both works, okay? It depends your budget, it depends what you really want to do, because you have really good points with Nanopore is that sometimes you have reads that could reach 200 KB, 300 KB. This is not what you have when you use PacBio, okay? So you have this advantage of having very long reads. And if you are interested in, for example, centromere evolution or something like this, it could be a good point. Okay, so it has advantages that you should not forget.
So I'm going to continue on this line of questioning because there's a lot coming in regarding this. Do you think that a hybrid assembly would improve the accuracy over a nanopore only contig?
Hybrid, you mean using both PacBio and Nanopore?
Probably because you had Illumina as well, so.
Illumina was absolutely necessary for the polishing step of the nanopore assembly, because even after the assembly, in the assembly just based on nanopore reads, we still had an error rate that is too high to publish, to deliver. Okay. For PacBio, it's different. With PacBio, you get hi-fi reads that are already more than 99% accurate. Then you do the assembly and you reach a level of accuracy that is probably closer to 200%. So I cannot say that you need Illumina when you do PacBio assembly.
Yeah. Okay. So which software did you use for the whole genome alignment and visualization? I assume that's your pipeline.
Probably it's about the second part of my talk when I compare genomes and transposons. Okay, so it's all made. Okay, the alignment they are based on BLAST. Okay, it's BLAST alignments, but we split the genomes by intergenic regions. So if you have 30,000 genes, you have 30,000 intergenic regions that are orthologous between all these genomes. And we just use BLAST region by region. Okay, there's no pipeline, I would say. Okay, it's just data management, parallel computing of thousands of regions. And the good point is that a transposon generally is not repeated many times within a single region, within a single intergenic space. Okay, it's not a repeated sequence, so you can do BLAST alignment of 100 kb versus 100 kb. Your alignment is easy to pass because you can easily retrieve what's specific and what's conserved. Okay, it's just playing with coordinates, so there's no pipeline completely. It's
process, perhaps.
Oh, yeah, it's all process exactly. That's what bioinformaticians use to do. And in terms of tools, because I've presented maybe this representation. Okay, so it's a very old viewer that I was using when I was a PhD student. It's called Rtbs comparison tool. It's because it's the only one I know how to use. Okay, it allows you to represent a BLAST, a full BLAST against two nucleotide sequences. So here I did this on 30 megabases. You see, so it's a very, very big BLAST. Okay, you have things similar everywhere, but you can zoom on a particular region like this one. So for people who wants to do that, it's not really easy to use. But if it's very big genomes, it's not easy, but it's feasible. And to answer the question, it's Rtbs comparison tool. But I don't know if there are now more modern tools to navigate in between two genomes, but I like this one.
All right, so we have time for just a few more, and I will say again, they'll all be answered in writing, so that will happen subsequently. So don't worry that your question did not get answered or asked live. Do you see that genetic maps will play a role in future assembly projects for pseudomolecule construction, for example?
Bionano maps. If I would plan now to assemble the wheat genome sequence, I would probably recommend to do Bionano maps, because even if you have a few hundred contigs or scaffolds, you want to work with 21 chromosome sequences. Okay, now we are in the area where we want to have one sequence per chromosome, and the best way to reach that is to add Bionano maps. It's easy to do, it's cheap, and high speed to avoid guiding the assembly based on a reference, because you can always have inversions, translocations that you won't see if you do not have IC. But I say that now because now the assemblies I can see are assemblies where you need a map, you need a Bionano map to go further, but maybe in a year or two, you use PacBio, you get 21 contigs, and that's done. So we are very close to that, actually. Close to having read assembly, where at the end you just get even 2 contigs per chromosome. That's okay. You don't need a map.
Yeah. So one of the questions that came in early was, if the goal is to have more sequenced genomes than Chinese Spring to get an idea about a pangenome, is it not a problem to base the assembly and annotation of Renan on Chinese Spring?
That's a very good question. And there are 2 questions, assembly and annotation. So first, the assembly is not really based on Chinese Spring. Okay, the assembly is done de novo, and at one moment we guided the ordering of scaffolds based on Chinese Spring, and then corrected what is wrong with Hi-C data, and Hi-C is based on Renan genome sequencing. So you can be confident that if an inversion, if you see an inversion, it's not because you made a mistake. Okay. So I would say no, it's not a problem at one moment to try to help you with Chinese Spring based on the synteny with Chinese Spring in the workflow to reach 21 pseudomolecules. That's not a problem. But for the annotation problem, is it a problem to base on Chinese Spring to annotate Renan? Of course it is, because and I told it, it's the reference annotation of Chinese Spring is probably full of mistakes. Okay, it's so complicated to predict genes just without manual corrections. Okay, that is probably full of mistakes, and we propagate these wrongly annotated genes in other genomes, and that's a problem. But for pangenomics, when you want to compare just your genes that are present or absent or in copy number variations, I think it's the only way to have something clean is to have a reference annotation. You try to identify the orthologs in the other genotypes, and then you say, okay, I have 2% genes missing, 3% genes missing. If you do de novo annotation of all these genomes, obviously we'll have just a mess, okay? So that's why it's a compromise between, okay, we have a reference annotation that has mistake, but we want to have a pangenome. So my idea is to take this annotation as it is. If we want to improve it, okay, let's go to improve it, but we will need to use this annotation to transfer to other genotypes, okay, and restrict the de novo gene predictions only to regions of the genome of Renan that is absent from Chinese Spring. This is what we did. Okay, I think it's the best way to advance towards a pangenome, because yeah, pangenomes based on de novo annotation of tens of genomes, it would be just a nightmare.
Well, I guess in some regard, you're basically trying to make sure you're comparing apples to apples, and you're not creating different, completely different references along the way. And but getting to this issue of, yes, we know that there's probably lots of errors in the Chinese Spring annotation. You at Clermont are actually trying to improve that with incorporating manual annotation into the process. So we would expect to see future releases, so to speak, that actually tried to correct that. And and do it and correct it in a manually curated manner. So,
Yeah.
so that we at least over time hope to have a much more accurate annotation of Chinese spring. So,
Absolutely, yes, they are. Yeah. We need to spend time looking at the genes, correcting the genes.
right.
But even manual curation, sometimes, even by hands, you cannot really decide if
Right.
this is a gene, this is the correct structure. Sometimes you just have not enough information, neither for an algorithms nor for a human to take a good
Right.
decision. So, yeah.
Well, that kind of leads me to my last question, which is in the vernalization gene A1 was only one copy was found in the Renan sequence. Is it possible that this was collapsed, the other copies, which we would have expected 3, that they were collapsed during the assembly?
Okay, I don't know I don't know exactly this this case of multiple copies of the VRN genes. Is it possible that it collapsed in the Renan genome assembly? I would say no. It's probably not collapsed in the assembly because I didn't see any cases where gene in tandem repeats like the glutenin genes were collapsed. Okay, it was clean. So I would say it's probably not collapsed, but if people didn't find a gene they expect to see in the annotation, it's probably because we had a problem with annotating the gene, okay? And it's probably because we projected the genes from Chinese spring to Renan. And I don't know if it's the case, but if you have several copies in Renan and one in Chinese spring, we probably just annotating this one, okay? And probably missed it when we do de novo annotation of the specific regions. Maybe this is why, but I will have a look at this particular case. No.
Yeah, so we may see that in other genes as well. So, yeah. All right, well, unfortunately, we're out of time, although we have a whole slew of great questions that have come in. But I do appreciate your taking the time to present the Renan genome sequence and annotation, and particularly what you've discovered on the transposable elements. And of course, I have my own questions about all of that. How this all compares with other crops and other plants. So, but in any case, that we'll save that for another day. But thank you very much, Fred, for your presentation.
Thank you, Kelly.
Thank you to all of our participants for some really great questions that came in. And again, we will get to them. And you'll find the answers in writing on the YouTube, IWGSC YouTube channel, as well as I think links to on our website. And we look forward to seeing you in November at our next webinar. So thanks everyone and have a great day.
Bye-bye.